Hi everybody! Today we're talking about the regulation of metabolic pathways, which is chapter 7 in your book. When we talk about cellular regulation, we can break this down into two parts, either regulation of gene expression or the regulation of metabolic pathways. This is something that many of you may be familiar with if you've taken or are currently taking biochemistry. We're going to start off by talking about regulation of gene expression. And a really great example of that is the regulation of dioxic growth. Dioxic growth is cell growth characterized by two growth phases. So let's take a look at this growth curve over here. And you can see it looks a little bit different from the growth curve we've talked about previously. On the left y-axis here, you see optical density versus time on the x-axis. These cells have both glucose and galactose in the medium. And what you're looking at here on the right-hand y-axis is how much glucose or galactose is left in the medium. So the first growth phase is shown by bacteria growth on glucose, which is the simplest and always the preferred sugar or carbon source. So you have your traditional lag phase here, followed by an exponential phase. And you see, as these cells are growing in that very first exponential phase, that the glucose is being depleted from the media. However, the galactose, which is the second or the non-preferred sugar source, remains constant in the media. The cells are going to enter a stationary phase here, then a second lag phase as they prepare to grow now on galactose because they have exhausted or used up all of their glucose. The second growth phase is then characterized by bacterial growth on galactose. So you can see as they enter this second exponential phase that the galactose is now being depleted from the media. So the regulation of dioxic growth is due to regulation of gene expression. In this case, it's due to the phosphotransferase system or the PTS system. This is a type of group translocation transport system where carbohydrate phosphorylation is coupled to carbohydrate transport. So in this case, we're going to be talking about glucose outside of the cell being in a non-phosphorylated state, and as it's brought into the cell, it becomes phosphorylated, which is represented by this P. Okay, so let's take a closer look at the phosphotransferase system and how it regulates dioxic growth. Okay, so the first step, and all of these steps, is when glucose is present in the media. So the first step is that a phosphate group is donated by PEP to HPR. So we'll talk about PEP conversion to pyruvate next week when we get into central metabolism, but this is the very last step of glycolysis. So PEP is going to lose a phosphoryl group to be converted to pyruvate. That phosphoryl group is donated to this small protein HPR. So HPR is now phosphorylated. HPR then is going to transfer that phosphate group to enzyme 2. Enzyme 2 is the glucose transporter, so you can see it here in the membrane. However, it also has a cytosolic component, enzyme 2A. So enzyme 2A can come interact with HPR, take the phosphate group from HPR phosphate, returning it to HPR, and then go back to the membrane to interact with the rest of the enzyme 2 transporter. If glucose is present in the media, as glucose is brought from the outside of the cell to the inside of the cell, it takes the phosphate group from enzyme 2A to become glucose 6-phosphate. So again, glucose on the outside of the cell is non-phosphorylated, however, glucose inside of the cell is phosphorylated. So now let's look at what happens if glucose is not present. HPR is still going to take that phosphate group from PEP. Enzyme 2A is still going to take that phosphate group from HPR, generating enzyme 2A in a phosphorylated state. If glucose is not present, so enzyme 2A cannot donate that phosphate group, it can instead activate the enzyme adenylate cyclase, right here snuggled up next to the membrane. Adenylate cyclase is an enzyme that converts ATP to cyclic AMP. And remember, this occurs because glucose is not present, and therefore enzyme 2A cannot donate that phosphoryl group, so it remains in a phosphorylated state. If glucose is present, this goes back to a non-phosphorylated state because glucose took that phosphate group. So when glucose is high, there is low cyclic AMP because adenylate cyclase is not being activated, again, because enzyme 2A will be in a non-phosphorylated state. However, if there's no glucose or low glucose, enzyme 2A in a phosphorylated state will accumulate. It can activate adenylate cyclase, giving us high levels of cyclic AMP. Cyclic AMP is an important co-activator in catabolite repression. We all learned catabolite repression in terms of the LAC operon. However, catabolite repression functions for many operons, LAC, but also all other alternative sugars, including maltose and galactose, for example. And these operons involved in metabolizing these alternative sugars are regulated by the presence of glucose. So the presence of glucose specifically represses the LAC and other genes responsible for the metabolism of alternative sugars because a better sugar source, in this case glucose, is available. So the cyclic AMP-CAP complex function together to activate the expression of lactose and again other sugar operons related to alternative sugar metabolism. CAP is the catabolite activator protein, and it requires co-activator cyclic AMP in order to again activate these expression of these operons related to alternative sugar metabolism. So if we look at, again, lactose as an example, however, this will work similarly for the maltose or the galactose operon. CAP bound to cyclic AMP can bind to an upstream site of the promoter, recruit RNA polymerase to the promoter, resulting in the expression of the LAC operon. So only when cyclic AMP is present, which if you remember only happens when glucose is not present, will the LAC operon and again the maltose and the galactose operon be activated. All right, so quick concept quiz. What would be the consequence of having a null mutation in the adenylate cyclase enzyme? Remember, that is this enzyme right up here. So remember, a null mutation implies that that enzyme is no longer present in the cell. So if adenylate cyclase is not present in the cell, it could never, under any condition, generate cyclic AMP. So if it's not generating cyclic AMP, it's going to have a difficult time metabolizing alternative sugar sources. So in this case, the correct answer is C. Cells would have a difficult time growing on lactose as the sole carbon source. Again, because it will be unable to activate the expression of the LAC operon, which is necessary to metabolize lactose. Let's do a second concept quiz. If you add bacteria to a culture that has both glucose and lactose, how will the bacteria ensure that glucose is utilized first? So remember, glucose actively represses the expression of the LAC operon because it doesn't allow for the expression of cyclic AMP. However, when glucose is absent, cyclic AMP can be generated and then allow the LAC operon to be expressed. So in this case, the correct answer is B, glucose inhibits the expression of the LAC transporter. For those of you who don't know what the term allosteric means, we're about to get into that right now as we talk about the regulation of metabolic pathways. So if we think about a cell has thousands of chemical reactions, and if they're not regulated, chaos can occur. The regulation of metabolic pathways is accomplished by adjusting the rate of one or more regulatory enzymes that control the overall rate of one pathway. So there are two general ways in which this can happen, either non-covalent binding between an enzyme and a pathway intermediate, These are typically allosteric effectors and will be the focus of the rest of today's lecture. There's also regulation by covalent modification, and I'll mention this at the very end, but again, our focus of today is going to be on allosteric regulation. So let's talk about patterns of regulation. Shown here in this diagram, this is a picture of an example metabolic pathway. So A can go through a series of reactions to be converted to either F or M or J, for example. So if we look at feedback or end product inhibition of a biosynthetic pathway, this is where the end product is a negative allosteric regulator. So you can see here, enzyme 2 takes B and converts it to G. G is then converted to H, which is converted to I, which is converted to J, which is the end of that pathway. J can then go back and inhibit enzyme 2. This is essentially saying, hey, you've already made this product, you can slow down or stop this reaction from happening. Another type of example is positive or feed-forward regulation. This can happen by an intermediate in a second pathway. So you can see here, K and G combine together to make L. So what K is doing, K is activating enzyme 2, saying, hurry up, because I'm ready, so I need G to be ready too. Another example of positive or feed-forward regulation is precursor activation. An example of this is G then activating enzyme 3, which converts the last, or catalyzes the last step in this process. Takes I and converts it to J. So what this is saying is, hey, this pathway is started, you better be ready there at the end. So G is going to come and activate 3 so that once I is produced, this last step can happen rapidly. We'll talk more about this a little bit later, but within any biosynthetic pathway, you're going to have both irreversible and reversible reactions. Reversible reactions, again, can interchangeably go forward and reverse. However, irreversible reactions can only move in one direction, but not the other. And regulatory enzymes typically catalyze irreversible reactions, many of which occur at branch points. So if we look a little bit deeper at patterns of feedback inhibition, we see here in this example from your book, 3 different patterns of feedback inhibition. In example A, you have simple feedback. So enzyme 1 converts A to B, enzyme 2 converts B to C. C can come back and inhibit enzyme 1. If we look at B, this is cumulative feedback. So A gets converted to B, enzyme 2 converts B to C. Now you have enzyme 3 and enzyme 4, and they each convert C to either D or E. For cumulative feedback, D can come back and inhibit enzyme 1, but so can E. And unless they are both bound to enzyme 1, you don't really get full repression. So both effectors need to be present to stop this pathway. Otherwise, you get a partial inhibition. If we look at example C here, we see an example of concerted feedback. So this is no inhibition at all unless both effectors bind. So now both D and E have to be bound to see any repression, whereas here each individually did some repression. However, you needed both for full. Here now you need both for any sort of repression. So this is an example of inhibition by isozymes. So if you had 2 different enzymes, each of which are completely capable of converting A to B, you need to inhibit both enzymes to stop producing B. Okay, so now let's define allosteric regulation. This is the binding of an effector to a site on the protein other than the active site, causing a conformational change and altering the activity of that given protein or enzyme. Okay, so let's break this down a little bit more. So this square is your enzyme. Here it binds to a substrate and converts that substrate to a product. That's its enzymatic activity. Here you can see a positive effector is bound. So it is bound to a site that is not the active site. However, it causes the active site to be open, so now the substrate can bind. The substrate will then be converted into a product. However, this enzyme has both positive as well as negative regulators. So if a negative effector is bound, again, this is bound to a site that is not the active site, it causes a change in the active site. So now the active site is closed and the substrate is no longer capable of binding. So now the substrate will not be converted to a product so long as the negative regulator is bound. Only when the positive regulator is bound will that active site open up again and substrate can bind and be converted into product. So let's look at that in a different way. Let's look at this figure here. So here we have enzyme 1, looks a little bit more like Pac-Man. It binds to this substrate, and you can see this substrate here should fit really well into that active site. However, you have an allosteric site here. So it's different from the active site over here. It's on a different part of the protein. However, when that inhibitor binds, it results in a conformational change, and now that substrate can no longer fit into the enzyme. However, in this case, now you see the closed active site, and an activator can now bind to that allosteric site, opening up that active site so the substrate can now bind. Now that substrate fits and can be converted into a product. All right, so let's take a second to check your understanding. How do allosteric effectors work? The correct answer is B. They bind to a site other than the active site, resulting in a conformational change, specifically of the active site. So let's look a little bit more at patterns of regulation. So let's see, in this case, we have a substrate that binds to enzyme 1. Enzyme 1 converts this green triangle to a purple square. Enzyme 2 converts the purple square into this blue rectangle thing. Enzyme 3 then takes the blue rectangle and converts it into the end product, which is an orange square. That orange square can then now be an allosteric effector inhibiting enzyme 1 from ever functioning. So this is an example of negative regulation of a pathway. This is what we refer to as feedback inhibition or end product inhibition, where again the end product then comes back and inhibits an earlier step in the pathway. Another example is positive regulation of a pathway. So here you have 4 enzymes, enzyme A, B, C, and D, where A is the initial substrate and E is the final product. So again, an example of feedback inhibition is where the final product comes back and inhibits an earlier enzyme. However, feed-forward activation or precursor activation is where a substrate or an intermediate earlier in the pathway can then activate an enzyme downstream in the pathway. So in this case, A is going to come and bind to enzyme D, activating enzyme D, allowing it to bind to substrate D and convert it to final product E. As we talked about earlier in the lecture, this can also be by an intermediate of a second pathway. Regulatory enzymes typically control branch points of metabolic pathways, and these reactions are usually irreversible. And we'll talk a little bit more about reversible versus irreversible reactions later. Okay, so let's take a second to test your knowledge. Consider the following pathway, and this is a simple pathway, no branch points. You have 4 enzymes. Enzyme 1 converts A to B, enzyme 2 converts B to C, enzyme 3 takes C to D, and enzyme 4 takes D to our final product E. So take a minute to draw an example of both feed-forward stimulation as well as end product inhibition. Okay, so one example, and these are not the only examples that are possible. However, one example is of end product inhibition. This can be where E, the final product, can go back and inhibit enzyme 1. An example of feed-forward stimulation would be that the product B can now come and activate the last enzyme, or enzyme 4. Okay, so to put this into context, let's talk a little bit about the kinetics of solute uptake. So if you add a solute, we can talk about solutes in terms of either amino acids or sugars, but let's think about sugars in this case. You're going to add them at different concentrations to a series of bacterial suspensions and plot the initial rate of uptake into the cell as a function of the external concentration. And that will give you a graph that looks something like this. So S is the concentration of the solute, so less to more of a sugar, and then V is the rate of uptake, so how much of that sugar is being taken up into the cell. So we are going to assume that the only significant route for solute entry is limited by the number of transporters. So if you have a cell and it has 1, 2, 3, 4, 5 of these sugar transporters, for example, if you only have one sugar molecule, it's going to be taken up very rapidly. So the rate at which solute enters is directly proportional to the fraction of transporters that are occupied with solute. However, now as you add more sugars, again, now we have 5 transporters, 5 sugar molecules. So they should all be transported fairly rapidly into the cell. However, as the external concentration of sugar increases, the larger fraction of the transporters bound to solute, meaning more of your transporters are actively busy bringing in that sugar molecule. So the rate of transport increases to a Vmax. Once you've reached Vmax, that means all the transporters are occupied. So even though you have more sugar out in the media, all the transporters are occupied bringing in sugar. So you've reached your maximum rate of entry. So if we take a look at this plot a little closer, so we have the Vmax, so this is again the maximum rate at which sugar can be transported, meaning all transporters are occupied and bringing in sugar. You can then have 1/2 Vmax, which is half of what you've measured Vmax to be, and then Km. Km is the concentration of solute that produces 1/2 Vmax. So how much of the sugar or solute needs to be present to reach 1/2 of that maximal intake rate. Km is assumed to be a measure of affinity of a solute for the transporter. So it can range from 1 micromolar to hundreds of millimolar. So an enzyme with a high Km, so that would be maybe over here on this axis, has a low affinity for a substrate. So meaning you need more of that sugar to reach maximal Vmax or maximum import. However, an enzyme with a low Km over here, like is shown in this figure, means that that transporter binds very rapidly to that sugar and can bring it in quite fast. So you need a lower concentration of solute to bring in or reach that half maximal transport. So these types of kinetics are referred to as Michaelis-Menten kinetics, and this is true of non-regulatory enzymes. However, regulatory enzymes, the enzymes we've been talking about, or allosteric enzymes, are an exception to that Michaelis-Menten model and have sigmoidal kinetics. So the binding of one substrate molecule increases the affinity of the enzyme for a second substrate molecule, or can inhibit the affinity of an enzyme for a second substrate molecule, or decrease the rate of product formation. So in the case where it increases the rate, this is positive cooperativity. In the case where it decreases the rate, sorry, I have that right here, this is negative cooperativity. Okay, so let's now look at the kinetics for regulatory enzymes. You can see they have this S shape or sigmoidal curve. So this makes enzyme catalysis very sensitive to small changes in substrate levels or concentrations. So when the substrate concentration changes, when substrate concentrations are very small in comparison to the Km, or again, the affinity of that transporter for its solute. So if we go back and look at these allosteric enzymes, it's not just how well does this enzyme want to bind to substrate, it's is there a positive effector that allows the substrate to bind? If the positive effector is bound, then it can bind very rapidly to the substrate. However, if that positive effector is not bound, then it won't bind. Similarly, if a negative effector is bound, it won't bind. However, if that negative effector is not bound, it could quite possibly very readily bind to its substrate. So let's take a second to check your understanding. So which mechanism could be used to regulate an enzymatic reaction? So what we've talked about so far is we can regulate the expression of the enzyme. That was an example we gave of the phosphotransferase system. Or we can change the conformation of the active site via allosteric regulation. So the correct answer should be D, or A and C. So all of that was to go over allosteric regulation. Another type of regulation is covalent modification. So covalent modification occurs by the reversible attachment of chemical groups, for example, an acetyl group, a phosphate group, a methyl, adenyl, or uridyl group. And these can be used to activate or inhibit the protein. And we've talked about a couple of examples of these in class, and we will talk about more later in the semester. But we've talked previously about histidine protein kinases and phosphorylated response regulators, for example. So once a response regulator becomes phosphorylated, it's typically activated and can do its job. However, in a non-phosphorylated state, it's inactivated. Later in the semester, we will talk about chemotaxis proteins, and their enzymatic activity can be changed via a methylation group. Okay, so the objectives for today were to understand the regulation of dioxic growth. An example of gene regulation by gene expression, particularly regulation by the phosphotransferase system. So understand the components involved and how they work together. So enzyme 2A, adenylate cyclase, and cyclic AMP, how they affect the expression of different sugar transporters. You should also be familiar with the regulation of metabolic pathways. So be able to define allosteric regulation, as well as feedback inhibition, feed-forward stimulation. Be able to look at a Michaelis-Kenton plot, be able to define Vmax and Km, as well as what Km means for the cell. So with that, I will see you all next week. And if you have any questions, please feel free to email me, and don't forget to post on the discussion board.